Hi friends, welcome you all. In our previous lecture, we have started food microbiology, and in that, we have started introduction to food microbiology, and we have already covered so many bacteria that are present usually in food, and they causes intoxication or foodborne infections. In today's lecture, we'll see remaining bacteria. So in that we are going to start with micrococcus. So micrococcus occurs in a wide range of environments including water, dust and soil. Micrococci are gram-positive spherical cells ranging from about 0.5 to 3 micrometers in diameter and typically appear in tetrads. Micrococcus has a substantial cell wall which may comprises as much as 50% of cell mass. So some of the species of micrococcus such as Micrococcus luteus, Micrococcus roseus, which is red in color, produces yellow or pink colonies when grown on monatoid short agar. Okay. So micrococcus is generally thought to be saprophytic and commensal organism, though it can be opportunistic pathogen, particularly in host with immunocompromised system such as HIV patient. Okay. So then next Proteus. So since it belongs to the family Enterobacteriaceae, general characteristics are applied to on this genus. It is oxidase negative, but catalase and nitrate reductase positive. Three species that are Proteus vulgaris, Proteus mirabilis and Proteus penneri are opportunistic human pathogens, and Proteus includes pathogen responsible for many human urinary tract infections like Proteus mirabilis causes wound and UTI infections. Then most strains of Proteus mirabilis are sensitive to ampicillin as well as cephalosporin, and Proteus vulgaris is not sensitive to these antibiotics. However, this organism is isolated less often in laboratory and usually only targets immunosuppressed individuals. Okay. So this organism will not going to cause any infection in a healthy individual whose immunity is strong, but this organism will surely attack on the person who is already immunocompromised. So Proteus vulgaris occurs naturally in the intestine of human and wide variety of animals. It is present in manures, then soil and polluted waters. Proteus mirabilis, once attached to urinary tract infection, infects the kidney more commonly than E. coli, and Proteus mirabilis are often found as a free living organism in soil and water. So in short, this bacteria of genus Proteus is present in soil as well as in water as well as in some polluted environments, and it has different types of organism just like Proteus vulgaris, Proteus mirabilis and Proteus penneri, and these organisms they are mostly involved in urinary tract infection. Then next Propionibacterium species, and in that the organism mostly responsible for causing pathogenic infection in food due to food is Propionibacterium frundaci. Okay. Historically, Propionibacterium species are of interest because of their use as a dairy starters, especially in production of Swiss type cheese, and their ability to produce propionic acid during their growth. So what is the special thing regarding this bacteria as their ability to produce propionic acid during their growth. So the genus Propionibacterium is generally split into cutaneous and dairy groups. The dairy Propionibacterium species can also be isolated primarily from dairy foods and spillage. The species in dairy production includes Propionibacterium genesii, Propionibacterium acidipropenici and Propionibacterium thionicii, Propionibacterium fecundici. So Propionibacteria have a role in production of flavor compound in cheese by proteolysis of some proteins and propionic acid production. So these are important with respect to cheese production is concerned, due to the product they made after proteolysis and propionic acid, they alter or change the flavor, and it is quite different from initial flavor and texture as well as taste. So dairy strains of propionibacteria are autolytic under environmental conditions found in cheese and degrade peptides and amino acids that are present in cheese, and the dairy species offer an interesting opportunity as a novel probiotic organism, with most obvious advantage being that they are considered safe for ingestion. So besides other activities, propionibacterium also serve as a good probiotic bacteria, and it produces propionic acid, and this propionibacteria is helpful in production of cheese as well as other products. Then next species is Pedagococcus species. In that Pedagococcus pentosaccus, then Pedagococcus acidalicti. So these are two bacteria here we want to describe. Then Pedicococci comprises a group of bacteria that are economic importance in brewing as well as in food industries. Several species and strains of Pedicococci have been used as a standard culture in fermentation of vegetables as well as meat as well as sausage products, then fermented milk, and associated with development of flavor in cheddar and other related cheese varieties. Some of the strains form capsular material that causes beer to become ropey and viscous. So they are catalase negative and exhibit homolactic type of fermentation and produce optically inactive lactic acid, that is it produces both mixture of D as well as EL type, and they generally appear in tetrads. Okay. So this is overall information regarding Pedagococcus bacteria. Then next, Pseudomonas fluorescens. So Pseudomonas, we are all familiar with this Pseudomonas. Pseudomonas fluorescens is a common gram-negative rod shaped motile bacterium. The organism is psychrotropic in nature, and it grows even at refrigeration temperature also at 7 degrees Celsius. It has extremely versatile metabolism. It can be found in soil and in water, and it is obligate anaerobe. This is peculiar feature of Pseudomonas fluorescens is that it is obligate aerobe, but certain strains are capable of using nitrate instead of oxygen as a final electron acceptor during cellular respiration. So optimal temperature for growth of Pseudomonas fluorescens is 25 to 30 degrees Celsius, and it tests positive for oxidase. So Pseudomonas fluorescens is also Pseudomonas fluorescens is also a non-saccharolytic organism. Heat stable lipases and proteases are produced by Pseudomonas fluorescens and other similar Pseudomonas. Okay, so these enzymes cause milk to spoil. So basically, Pseudomonas involved in spoilage of milk by producing some enzymes, and that enzymes causes spoilage in milk, and these enzymes are categorized in the group of lipases as well as proteases, where these enzymes when they enter through milk into our body, they causes degradation of our proteins and other lipid molecules. Okay. So these enzymes cause milk to spoil by causing bitterness. So due to spoilage of Pseudomonas fluorescens, the taste of milk is look like somewhat bitter. Then it causes breakdown of casein and ropinase due to production of slime as well as coagulation of proteins. So this is how Pseudomonas fluorescens affects milk. Then next, Pseudomonas aeruginesa. So again it is common Pseudomonas organism. It is gram negative aerobic rod shaped bacterium with unipolar motility. It is an opportunistic human pathogen. What is meant by opportunistic human pathogen is that so when your immune system get compromised due to some reasons, then normal flora of your body they get activated and they cause illness otherwise these normal flora are they don't do anything, though they are present in your body or within your body, they don't do anything, they are helpful for us, but in a opportunistic condition or in some immunity compromised situation, these opportunistic pathogen get activated and they causes diseases. So Pseudomonas aeruginosa is also a opportunistic pathogen in plants also. Pseudomonas aeruginosa is a type of species of genus Pseudomonas, that is Migula. A gram-stained Pseudomonas aeruginosa bacteria, which is look like pink red rods, secrete a variety of pigments including phycocyanin, which appears to be blue green, then pyoviridin, which appears yellow green and fluorescent, and pyruvin, which appears as a red brown. That's why we called as this is fluorescence, because it produces some fluorescence or some color compound after putting into some media. So Pseudomonas aeruginosa is often preliminarily identified by its fluorescence and a grape-like or tortilla-like odor in vitro. Then definitive clinical identification of Pseudomonas aeruginosa often includes identification of production of phycocyanin as well as fluorescein. So production of these two compounds we can use as a test for identification of Pseudomonas aeruginosa from rest of the Pseudomonas species. So as well as its ability to grow at 42 degrees Celsius. So these are some peculiar features of Pseudomonas aeruginosa, that it produces pigments like phycocyanin, then fluorescein, as well as it can grow at temperature even at 42 degrees Celsius. Then Pseudomonas aeruginosa is capable of growth in diesel and jet fuel also. Okay, this is again peculiar thing in case of Pseudomonas aeruginosa, that it is capable of growing in diesel and jet fuel, where it is known as hydrocarbon using microorganisms or humbugs. So what is the term used to denote organisms that are utilize hydrocarbon as their food? They are denoted as humbugs. So this causes microbial corrosion, and Pseudomonas aeruginosa is considered by many facultative anaerobe. Okay, so this is a picture that is showing stain preparation of Pseudomonas aeruginosa. The next important group of bacteria is Salmonella, and in that Salmonella typhimurium, then Salmonella typhi and Salmonella centroides. So these are again important pathogen from this group which are usually present in food. So Salmonella species have been reported to be a leading cause of foodborne illness in humans. So this is one of the major foodborne illness that we face. So foodborne salmonellosis scores over all other foodborne bacterial illness in humans. So this is the main foodborne illness, that is most of the people suffer from this illness after ingestion of spoiled food, and it is the leading disease that is caused due to consumption of spoiled food. So Salmonella species are mainly responsible, and they are majorly food spoilage causing bacteria. So it has been reported to be a leading cause of foodborne illness in humans, and foodborne salmonellosis scores over all other foodborne bacterial illness in humans. So enteric fever is a serious human disease associated with typhoid and paratyphoid strains. Okay, so you are all familiar with the typhoid as well as paratyphoid. What are the symptoms and what are the clinical complications you have studied in medical microbiology. Here we will not going to discuss lot much thing about that, but you have to just check which of the pathogens they are present in food. So Salmonella enterobacteriaceae. So these are two strains of typhoid and paratyphoid strains. This includes Salmonella and enterobacteriaceae. The optimum growth temperature is for these bacteria is 37 to 45 degrees Celsius. The organism can also grow at about 7 degrees Celsius in food also. Even so, this you can see that even in refrigerated foods you can found that Salmonella may be present at refrigerated foods. It ferment carbohydrate with production of acid and gas. Okay, so this is fermentation with acid and gas, and Salmonella are oxidase negative, then catalase positive, and grow on citrate as a sole carbon source and produce H2S. Okay, some of the Salmonella strains can grow at higher temperature even at 54 degrees Celsius, while others exhibit cyclotropic properties. So, the organism has ability to grow at pH values ranging from 4.9 to 4.5 to 9.5. This is quite wider pH range with an optimum pH growth at 6.5 to 7.5 per species. So, some species are facultatively anaerobe. Okay, there are some species in this Salmonella group, they are facultatively anaerobe. Small gram negative non-spore forming rod shaped bacteria. These are measured in size, that is 2.2 to 4 millimeter, and they are belonging to this family. So, milk as well as meat and poultry are principal vehicle of human foodborne salmonellosis. So, salmonellosis is the disease in case of food we got from foodborne infection. So, these are the three sources that are milk, then meat, and some of the poultry product. They carry this Salmonella bacteria, and consumption of this product, if they are infected with this Salmonella species of bacteria, we definitely got salmonellosis. Okay, so these are the three important sources from we got infection of Salmonella species. So, ingestion of only a few Salmonella cells can be infectious. So, low levels of Salmonella in finished product may therefore be serious public health consequences. So, this is the main foodborne pathogen as far as other bacteria are concerned. So, this is how Salmonella look like. In this picture you can see that. And then next bacteria is Serratia. So, Serratia is a genus of gram negative facultative anaerobic rod shaped bacteria which is belongs to Enterobacteriaceae family, and the most common species in the genus Serratia marcescens. It is normally the only pathogen and usually causes nosocomial infection. Nosocomial infections means the infections which we acquire from hospitals. So, these are categorized under nosocomial infections, and these species of Serratia, they are mostly involved in nosocomial infection, but in food also they are present and they cause foodborne illness. So, however, rare strains of Serratia of so these are different strains here mentioned, and these are cause disease through infection. So, members of this genus produces characteristic red pigment and proteogeosin. Okay. So, this is overall information about Serratia. So, basically it is very less involved in foodborne infection, but still it is present in food. Some foods sometimes it get infected, and mostly this bacteria is involved in nosocomial type of infection. Okay. Then next is Streptococcus thermophilus. So, this is the only species of Streptococcus that is associated with food technology. Okay. So, the only Streptococcus species that is associated with food technology or food is Streptococcus thermophilus, which is used in the manufacturing of yogurt in co-culture with Lactobacillus bulgaricus and dahi. So, again this is a beneficial activity of Streptococcus thermophilus, that it is used in fermentation of dahi and other products. So, Streptococcus thermophilus is basically a gram positive facultative anaerobe, and it belongs to family Streptococcaceae. It is a catalase negative organism that is non-motile. it does not form any kind of spore, and it is homofermentative and occurs in pairs to long chain. Its shape is spherical to ovoid with diameter that is range from 0.7 to 0.9 micrometer. The optimum temperature for growth of this organism is between 39 degrees Celsius to 45 degrees Celsius. Although most species in the genus are able to grow at temperature ranging from 45 to 60 degrees Celsius, they do not grow at temperature below 20 degrees Celsius. Okay, but they can survive even at 65 degree for 30 minutes. So this is the pasteurization temperature. So this Streptococcus thermophilus, they still can survive after pasteurization. So they ferment sugars with lactic acid as a major end product and produces around 0.6 to 0.8% lactic acid. So that's why these bacteria, Streptococcus thermophilus, they are used in the fermentation of milk along with lactic acid bacteria in production of some product like cheese and dahi. Then they are able to grow both in 2.5% salt solution, that is NaCl solution, but fail to grow at 6.5% NaCl at pH 9.5, 9.6 or in a milk with 0.1% methylene blue. Okay. So these are all data according to Burgess manual, and it is also classified as a lactic acid bacteria, that is LAB, and it is very versatile organism. Why it is called as lactic acid bacteria? Because it produces lactic acid after fermentation of sugar lactose, and it is present in milk. That's why it is categorized under lactic acid bacteria, which is designated as LAB or lab. It is very versatile organism. So Streptococcus thermophilus has properties that make it one of the commercially most important lactic acid producing organism. Okay. Then Streptococcus thermophilus is used along with lactobacillus species as a starter culture to manufacture several important fermented dairy products including yogurt and mozzarella cheese. So in this way, this is the basic information regarding Streptococcus thermophilus, and it is the main lactic acid bacteria which is used along with the lactobacillus species, and both of these bacteria they use as a starter culture for production and manufacturing of different products which are derived from milk. Though the natural habit of Streptococcus thermophilus is yet to be established, the most strains have been isolated from milk environment. So the basic environment for this bacteria is milk. So if you want to isolate that bacteria, so you have to take a milk sample. So milk is the principal environment, basic environment for this Streptococcus thermophilus bacteria. Okay. The next again important pathogen is Staphylococcus aureus. Again, this is common bacteria. You are all heard about this bacteria. This Staphylococcus aureus is commonly associated with humans. It is a normal flora. It is present as a normal member of normal flora of your body. It is gram positive, catalase positive cocci. Staphylococcus aureus is a common cause of foodborne gastroenteritis known as staphylococcal food poisoning. Okay, so the disease mainly caused due to spoilage of food by the Staphylococcus aureus. It is known as gastroenteritis and it is known as staphylococcal food poisoning. So staphylococcal gastroenteritis is caused by ingestion of food that contains one or more enterotoxin which are produced by some of the strains of Staphylococcus aureus. So basically this bacteria cause infection of foodborne illness because it produces a toxin which is categorized under enterotoxin. Enterotoxin means toxin that act on your stomach, and after ingestion of such a food which is contaminated with this kind of toxin, the person is suffer with gastroenteritis, or it causes gastroenteritis. Okay. So although enterotoxin production is believed generally to be associated with coagulase and thermonuclease producing staphylococcus strains, many species of staphylococcus that produces either coagulase nor teenage are also known to produce enterotoxin. So there are many strains of Staphylococcus aureus that they produce this enterotoxin, and they causes gastroenteritis in a person who consumes that food which is contaminated with this bacteria. And the main reservoir of Staphylococcus aureus is nasal cavity of human beings, from where they find their way to the skin and bones. So mainly this bacteria in your body, which it is present in the nasal cavity, and they can establish skin as well as wound infection when they got favorable and suitable conditions. So mastitis in animals due to Staphylococcus aureus is quite common, and from infected udder the organism finds its way into milk. So in milk this organism basically comes from the diseased udder. If that animal is suffered with mastitis, or and we drop milk from that such an animal, then definitely it contains Staphylococcus aureus, and it get contaminates the milk. Then this organism can grow with well in NaCl concentration. So this is again peculiar thing in that it can tolerate high salt concentration up to 7 to 10%, though the optimum growth of temperature for this organism is 37 degrees Celsius. This optimum growth for this organism is 37 degrees Celsius. So that's why this is a normal member of normal flora of your body, and it is commonly found in our body, and some strains can grow at temperature at low as below 6.7 degrees Celsius. So this organism can grow in water as well as water activity as low as 0.86%. So this is overall information regarding Staphylococcus aureus. So basically this organism cause foodborne infection by producing some toxin that are known as enterotoxin, and it causes gastroenteritis. Okay. The next again important organism is Shigella, which causes Shigella dysentery. You heard this term, that is Shigella dysentery. And bacillary dysentery or shigellosis is the common term used to denote Shigella dysentery, and it is caused by Shigella species. So Shigella is a member of a family which this member belongs to family Enterobacteriaceae, and the growth temperature varies from 10 to 48 degrees Celsius. Shigella does not usually survive well in low pH foods. Okay, so this bacteria again one peculiar thing in case of this bacteria is that it cannot survive in acidic foods. The foods which has lower pH, the chances of survival of this survival of these bacteria are very few. So Shigella is sensitive to ionizing radiations. So species are non-motile, oxidase negative, producing acid, not only acid, not only gas from sugars, and they do not grow on citrate as a sole source of carbon, and do not grow on Casein agar, and unlike Salmonella they do not produce hydrogen. So these are some biochemical peculiar features of this Shigella species. And shigellosis is an important disease in developed and developing countries. The disease is caused by ingestion of contaminated food, and in some instances it subsequently leads to rapid dissemination through contaminated feces and infected individuals. So this bacteria, as it belongs to Enterobacteriaceae family, so again we can form this, it is categorized under coliform, and it may enter through fecal contamination, and it causes shigellosis as well as Shigella dysentery, and the infective dose may be low, below as per means below 100 cells. And the contamination of foods usually do not occur at a processing plant, but rather it occurs through an infected food handler. Okay, the humans are a natural reservoir of Shigella. Basically this bacteria is present in fecus. So humans are the main reservoir of Shigella, and the organism is spread through fecal oral route. So if the fecal matter, if it get mixed in water, especially drinking water, so there are chances that the shigellosis chances. Means the bacteria will enter into that drinking water, and chances of shigellosis are much more highly. Then next is Vibrio. So Vibrio cholerae and Vibrio parahemolyticus are the two important species of genus Vibrio. So Vibrio cholerae causes cholera, and one of the few foodborne illness with epidemic as well as pandemic potential. So Vibrio cholerae are gram-negative straight and curved rods, and they belong to the family that is Vibrionaceae. An important distinction within species are made on the basis of production of cholera toxin. So it is again an important disease that is caused by ingestion of contaminated food, and in that we are all know that cholera it is a disease that is caused by Vibrio cholerae, and this particular disease of cholera it is caused due to an enterotoxin, that is CT, produced by this Vibrio group of bacteria. So Vibrio cholerae is a part of normal free-living bacterial flora in estuarine areas. Among the many different estuaries, estuaries broth describe the isolation of Vibrios in alkaline peptone water and is mostly commonly used. Though Vibrio parahemolyticus can grow in presence of 1.8% NaCl, the best growth occurs in the salt concentration that is maintained at 2 to 4%. So again, this is important foodborne pathogen, food contaminating bacteria and pathogen, that is Vibrio. So this is how Vibrio look like. It is look like you can see in this picture. Then next organism is Yersinia. So Yersinia enterocolitica or Yersinia pestis are the two important human pathogens. So while Yersinia enterocolitica causes foodborne gastroenteritis, Yersinia pestis is an agent of human plague. Okay, so this is you might be heard about this term. Bacteria, that is Yersinia pestis, it is a causative agent of human plague, and Yersinia enterocolitica, it is caused gastroenteritis. So both of these bacteria, they belong to genus Yersinia, and Yersinia enterocolitica, also known as newly emerging human pathogen. So it is new. For us, it is newly emerging human pathogen, and it is a heterogeneous species that is divisible into a large number of subgroups. So Yersinia enterocolitica is unusual because it can grow at temperature below 4 degrees Celsius. So you can see that even at freezing temperature, this bacteria can easily grow. So this is one peculiar thing regarding Yersinia enterocolitica, is that it can still may present in frozen foods, and the general generation time at the 28 to 33 degrees Celsius, which is optimum growth temperature for this, is almost 34 minutes. It also survives in frozen foods, as I have told you before. So it grows better in processed foods such as pasteurized milk, then vacuum packed meat, as well as boiled eggs, as well as boiled fish and cottage cheese. So both these species can grow over pH range of 4 to 10. So optimum pH range for this bacteria is 7.6, and it can tolerate alkaline environment as well. They can motile at a temperature at greater than 30 degrees Celsius. However, both these organisms are susceptible to pasteurization, ionizing and UV radiation. So the organism can also tolerate salt concentration which is maintained to 5%, and infections with Yersinia species are due to transmittance of this organism from animal to humans, and organism is frequently present in pork, lamb, poultry and dairy products. So basically this organism is present in poultry as well as meat product, and this causes again severe infections after ingestion of this bacteria contaminated food. So this is overall regarding detailed information regarding types of bacteria that are present in food and the illness they cause or the type of complication they cause. So in next lecture we'll going to see or we're going to study different types of fungi, yeast as well as mold and viruses they causes means these type of organism they present in food and how they causes different types of illnesses. So we'll see in next lecture. So in next lecture we're going to see yeast, mold and viruses and how they cause foodborne infection. Thank you.